Hello, good afternoon, everybody. My name is Andrea Sway. I'm a professor in the biology department, and I'm also the graduate student coordinator. And it was my crazy idea to try to get a bunch of people to give their research talks as grad students in 5-minute lightning talks. And so I want to thank these students, these 9 students that are on the board, for volunteering to speak today. This is a really hard format to present in, but it's also really fun to listen to. So we really appreciate them taking the time to do this. We have an amazing department with lots of great research going on, but sometimes it's hard to know what everyone is working on. And so this is a great opportunity to hear what people down the hall might be working on. And also a good way to showcase all the great research going on in our department as well. And so I don't want to take up too much time. We have 9 great talks to get through. They're going to be 5 minutes each. We're going to try to keep you on time. I think Kathy's going to give you a 1-minute warning at 4 minutes or so. So we'll try to keep things relatively on time. And then we'll do questions at the end. I think we'll start with faculty questions, and then all the faculty are dismissed, and then the students can take over and ask their questions. And we have people online as well, so I can help monitor the questions online as well, or maybe I'll try. Okay, anything else? No, I think that's it. So we'll go ahead and get started. We will be cycling through these different talks. And so we're starting with Celia Hodson from Jonathan Stillman's lab. And you have the floor in 5 minutes.
Do I hold it just like this? Okay. Hi, I'm Cecilia, or Cece. And like Andrea said, I'm in Dr. Stillman's lab, otherwise known as the Crab Lab. And I'm here to talk to you about my research on unraveling the genomic basis of heat and pH tolerance in 2 porcelain crabs. There you go. There we go. Okay. Okay, so starting out, I want to talk a little bit about the subjects of my research. I'm studying 2 crabs that are native to the Pacific coast of North America. They overlap in range along the Pacific coast, but they are segregated by tidal zone. The top species is Petroleuces syncytipes, and it exists in the intertidal zone, meaning it experiences regular high tide, low tide cycles. And the second species, or on the bottom, is Petroleuces manimaculus, which is subtidal, so it has very limited exposure to tidal cycles. And from previous research, we do know that the intertidal crabs are a lot more tolerant to environmental stressors when compared to its subtidal counterpart. So the question that my research revolves around is understanding the differences in this physiology and tolerance based on a genomic level. So specifically, how are the physiologic differences explained by genomic differences? My hypothesis, based on the known tolerances of the crabs, is that the intertidal crab experienced genomic changes that resulted in a better tolerance when compared to the subtidal crab. I'm using 3 major methods to explore this question. The first is understanding the differential gene expression, comparing the genes expressed between the crabs exposed to different stress treatments. So long-term stress versus short-term stress, and no stress versus stress treatments. The next is seeing what these differentially expressed genes are actually doing through functional annotation, or more simply, what biological function they are associated with. Third, I am comparing the locations of the differentially expressed genes in the genomes to understand better how the genomic changes may have impacted gene expression. Starting out, I want to show the differential gene expression results. These are some complicated plots that I feel like I don't have enough time to talk about, but starting out, these are 2 cluster plots showing the differential gene expression for the intertidal crab on the left and the subtidal on the right. And the columns are samples by treatment group, and the rows are individual genes. The little rectangles shown in red are the genes that are expressed more than the normal levels. So these genes here. And then the ones in blue are expressed less than the normal levels. I'm mostly interested in the red, as they show the genes that are just really active in the stress treatment groups. But the blue genes will provide some additional context as well. Firstly, looking at the plot, we see a few things that stand out. First, the heat-tolerant crab expresses more genes, with 499 being differentially expressed. Second, we see clear patterns of expression by treatment in the intertidal crab that we don't necessarily see as much of in the subtidal crab. And third, not every crab has equal expression patterns, indicating that there's some intraspecific variation in stress response. To compare the genes, I chose what's called an orthogroup, which is just a group of genes that originates from the same ancestral gene. The orthogroup that I chose is orthogroup 210, which is located right here in the short-term stress response of P. sanctipes. I chose this particular orthogroup because there are two genes in Petrolisthes sanctipes belonging to this orthogroup that are differentially expressed with short-term stress. But the two corresponding genes in the subtidal crab are not expressed at all. To look at the genomes, I made this plot showing where the genes are in relation to one another on each genome. Each line is a contig, which is just a segment of sequenced genomic DNA. They aren't chromosomes, they aren't representative of chromosomes, but they do show where the genes are in relation to one another. The blue stars show where the genes are, and I did label the distance in base pairs between the two genes. You can see that the genes in P. sanctipes are over 4,000 base pairs apart, whereas P. manamaculus have only 2,000 base pairs separating the genes. The question now is if the difference is the thing that is impacting the gene expression, or if there are other factors around the genes that may be impacting the expression, such as promoter regions or regulatory regions that may be upstream. There's also the possibility that it could be on different, like there could be regulation factors that exist on different chromosomes, but that is outside of the scale of my particular question. So the next steps are doing a better plot, essentially, to better understand how the regions around the genes align to one another. These are examples of synteny plots, which just shows how the two chromatids or how the two sequences really align to one another. And this gives us an idea of if there's insertions, if there's deletions, if there's rearrangements, or inversions, or any of the other fun stuff that chromosomes like to do over grand evolutionary scales. So I do want to keep talking about my project. I have a lot more to share and a lot more context, especially, but I do only have 5 minutes. So thank you to listening to my speed talk, and enjoy the rest of your talks.
I'll take 30 seconds to talk to a neighbor while I switch over. All right, Michael, let me cycle 2.
Sorry, guys.
No, you don't have to hold it. Like you just grab it right here. Yeah. Michael, all you're gonna do is
Like translate.
oh, you want to do?
No.
Oh, no, it's not on. It's just a video. It's being recorded.
Okay.
Yeah, and you're just gonna click on this button. Okay. All right, go ahead, Michael.
Hi, everyone. My name is Michael Acholonu, and my topic for is characterizing escape bacteria and phage association in effluent wastewater treatment plant. A little bit of quick background. I know people might be asking what is escape bacteria. Escape bacteria are pretty much antibiotic resistant bacteria that kind of create problems in clinically around the world. Escape is just an acronym for the names of the different bacteria, with the name being Nitrococcus pachymium, Staphylococcus aureus, Klebsiella pneumoniae, and so on. I don't want to call all the names over there because of time. But with them, they do come in different kind of morphological shapes, but those morphological shapes doesn't mean that they wouldn't be causing issues for the world as we know. So my thesis was based on two aims: to investigate the relative abundance and distribution of escape bacteria and influent wastewater treatment plant, and also to look at investigate phage association on those escape bacteria. Some of the methods that I kind of did was pretty much collecting those wastewater from the wastewater treatment plants and doing some DNA extractions on them. And after doing those DNA extractions, I happened to do some genomic analysis and assembly to be able to get results about my data. These were like a preliminary results visualization of what the prevalence of escape bacteria, with the first diagram showing us the prevalence of escape bacteria in those wastewater, in the influent wastewater when collected. And the phage association was showing the different phages that were, could be a little bit not clear, but the phage association was showing the different phages that were more prevalent in those escape bacteria that were had more phage interaction versus other escape bacteria. So the primary idea of conclusion of why I was doing this project is to be able to monitor wastewater in that way we can get a first idea on how to control antibiotic resistant microorganisms. And a next step towards this would be trying to investigate influent versus effluent. Influent backstory is the idea of raw sewage water versus effluent, which is cleaned up wastewater. And a secondary aspect of looking towards this phage association is to be able to understand the how environmental phages could be a potential treatment route, by like looking at phage therapy as an alternative means to antibiotics in terms of medications that they're about. And a quick acknowledgement to all the people around, like the SEO, my lab teams, and the phage pathway, which also sponsored me, and also my different advisors that kind of like helped me through this project.
Right, again, take 30 seconds to talk to a neighbor. Hi, Gabriela, how are you? Where's the chat? And then Gabriela, I'm gonna all you have to do is press this button to go forward. You get it, grab it here. Okay, no, yeah, and then you can stand here to make it. And I'll go like this and have 1 minute. All right, go ahead, Gabriela.
Hi, I'm Gabrielle. Oh, mic. Hi, I'm Gabriella. I'm a second-year master's student, and I'm going to talk to you guys about combating antimicrobial resistance with campus wastewater surveillance. First, I'd like to thank my lab, the Anand Lab, and SEO and NIH and Climate HQ for funding me. So, getting into it, So a bit of background, antimicrobial resistance is when the overuse of antibiotics in either clinical or agriculture settings leads to antimicrobial resistance, where microbes become resistant to antimicrobials. And this is a rising cause for concern, because deaths caused by MR are projected to reach 10 million people per year by 2025. Very concerning. And so wastewater-based epidemiology has historically been used to track things like polio, typhoid, pharmaceuticals, and like drugs and biomarkers, and many more. And it's been recently used to track COVID-19. You guys would know about that. So it's different to clinical sampling that just samples patients that would go to the hospital. It samples a wider population of just everyone contributing to wastewater. And so it's like a wider view than clinical sampling. And it can like what we heard about like asymptomatic and symptomatic patients, it can capture symptomatic patients. So now getting into my project's aims, I aimed to look at a couple different things. So one of them was microbial taxa in the wastewater microbiome at San Francisco State. And then I also aimed to look at AMR genes of the wastewater microbiome and seasonality of the taxa and AMR genes. And then also mitigation steps, which would be looking at phages that can infect these antimicrobial resistant bacteria. And so the way I was looking at these aims is by collecting wastewater at the SFSU campus at these manhole covers. And so the way I would do it is deploy tampons, and that would and then they would sit there for 24 hours, and that would give me a composite sample, which would be better, which is better than a grab sample that would just grab one time point. So it would be like it would get like the fluxes throughout the day. And then I would take this wastewater back to the lab, and we process it with nanotrap nano beads, and they're magnetic beads that bind to microbes. And then there's data analysis that's still ongoing to to get all those aims that I mentioned earlier. And then while this was going on, I was also doing protocol development for a protocol that would look at multi-level, the microbiome on a multi-level stage, and it would also be focused in mostly on bacteriophages. And so here we have some bacteriophages, and on the bottom you have some that were isolated from a wastewater treatment plant, but then up here the bigger one is a phage that was identified from the wastewater campus, wastewater from the San Francisco State campus. And thank you for listening.
All right, talk to your shoulder buddy for 30 seconds. So all you have to do is click this button to move forward, and then put the mic, grab the mic close to your mouth, and I can hear you. And then I'll be right there. What is it? All right, so now it's time for Jillian.
Hi, can you guys all hear me with the mask on?
Yes.
Okay, awesome. Hello, I'm Jillian Valencia, or Jill. I'm a master's student here at San Francisco State, and I'm in the Cell and Molecular Bio program. Thank you to Dr. Sway for inviting me today. I am in Dr. Jose de la Torre's laboratory, and we study thermophilic microorganisms. And so if you're wondering what thermophiles are, they're part of a larger group of extremophiles, and thermophiles are these heat-loving organisms. They inhabit a range of around 40 degrees Celsius to 122 degrees Celsius. I believe this is 105 Fahrenheit to 250 Fahrenheit, so really hot, hence the name thermophiles. And so we study extremophiles to inform us more about the limits of life. And this is a big thing that drew me to San Francisco State because I was really interested in astrobiology. I really wanted to know how early life forms were able to adapt and live in these really harsh conditions. And astrobiologists like myself, we commonly will study hydrothermal vents and geothermal springs as sites that are analogous to what Earth was like during the Archean eon, which is when life is thought to have formed for the first time. And so my questions very broadly are, how do natural populations change over time and who do we see here? And I want to emphasize natural populations because we're not isolating any microbes and culturing them in a lab because this would not be a natural population. So we really want to see what is going on at that specific time point. And so to do this, we're taking an entire environmental DNA sample and sequencing the entire thing and reconstructing it. And so I have a total of 10 sample points across the spring. A, B, and C I've shown here are sediment communities, and site W in the middle is a water column community. And throughout the year, the temperature fluctuates. And you can see that B and C, they fluctuate by 13 degrees Celsius, which is a lot. It fluctuates a lot overall. And so I have 10 sample points. That adds to 9, I'm sorry, like it's supposed to be 4 for June. And so my goal is to reconstruct the genomes of all 10 sample points, and then I would be able to analyze the differences in the community composition, who's there at that time point, and in what amounts. And so largely the results of this are from the 10 different independent samples, we were able to reconstruct 1,000 MAGs or metagenome assembled genomes. This is the fun part. 300 of these are novel species that have never been cultivated or identified. That's a lot, right? So that was really exciting for me. 260 of these, we know the genus that they're in, but at least 45 to find new genuses or higher level taxonomic groups. So a lot of these organisms we're seeing here have never been studied, never have been seen. No one knows anything about them, which is really exciting for me and my lab. And to answer one of my other questions about how do natural populations change over time, well, I could tell you for certain that they're changing. This is a abundance heat map. And so I have on the bottom here the different sample points, the 10 sample points. And along the y-axis is going to be 30 of the top mags that I'm finding across the sample sites. And my lab's bioinformatician, me, as I'm still analyzing the data, so I can't tell you with like statistical significance what's changing, but it is changing. So that's really exciting to know. And with that being said, thank you. I hate to run, but I am going to be leaving after this. So if you have any questions, that's my email. My PI is also here, so I'm sure he'd love to answer questions. And thank you to all these lovely people on the screen that have helped me along the way. Thank you.
So take 30 seconds to chat. Five. Okay.
2 questions.
Five. Is it Clara? Clara. Clara. Perfect. So I just gotta tell you 2 things to move forward. All you're gonna do is press this button here, and then you're gonna have the mic in the president, so that everyone can. And then I'll say this. Yeah. No one's going really over, so you'll be fine. Yeah, you'll be fine.
Okay.
All right, and then let me read. Wonderful. Okay, so next we have Clara. Thank you.
Hi, my name is Clara. I'm a graduate student in the lab of Dr. Joseph Chen here, and I'm going to talk to you a bit about some of my work identifying an atypical ABC transporter in the bacterium Centrorhizobium mellilodi. So Centrorhizobium mellilodi, it's a gram-negative bacterium. We use it as a model for investigating host microbe symbiosis, as well as generally investigating bacterial physiology. So Centrorhizobium is known for colonizing the roots of plants in order to form symbiosis. It does this by forming nodules that you can see here on the roots. The bacteria will then enter the nodules and perform nitrogen fixation, which will then provide bioavailable nitrogen to the plant. So this project actually originally got started during work trying to identify causes of my God, membrane defects. And what you can actually see here is we had this issue where we kept seeing weird unexplained differences in SDS sensitivity. So we have the strain here, Joe 6730 is constructed here, and then CL 150 is constructed in a collaborator's lab. And despite both coming from the same parent and supposedly being the same genotype, we're seeing a really big difference in sensitivity to the detergent SDS. And we kept seeing this dichotomy across a lot of different strains from different labs. So we ended up performing whole genome sequencing, and we identified the likely cause as an indel in this stretch of Cs down below. And this was in a gene called SMB21269. And if we went ahead and deleted that from our resistant parent, we're actually able to reproduce the sensitive phenotype, confirming to us that that was the cause. So we went ahead and did a screen for this deletion against a bunch of different antibiotics. And we found that it was causing increase in sensitivity to a wide range of them of all different kinds of mechanisms. Shown here is an example with chloramphenicol. Here is just growth curves, and you can see how as we increase the concentration of chloramphenicol, our resistant wild type is starting to outcompete our deletion mutant here. And this is obviously representative of other antibiotics as well. So SMB21269 encodes an ABC transporter or an ATP binding cassette. These are just kinds of active transporters using ATP to transport substrates across the membrane. The core of them consists of a pair of transmembrane domains and nucleotide binding domains, and they're found everywhere all across life. So shown here on the left is an example of a typical ABC transporter. This is MSBA from E. coli, has a single transmembrane domain and a single nucleotide binding domain, and then two of these proteins would come together to form the complete transporter. Here on the right, we have our transporter, which once again has those two domains, but it also has this extra accessory domain on the side. And bioinformatically, it appears that this domain is well conserved across related bacteria, but it's completely uncharacterized. And so we're just calling it DUF for domain of unknown function. And when you find something like this, the next thing you want to do is delete it. So we created a strain where we removed the vast majority of DUF, and then we can go back and do antibiotic sensitivity testing. And so right again back here, what we can see is our deletion is our orange here at the bottom. It's totally dead as we increase concentration. And then we can also see blue is our wild type, which is growing as before. But interestingly, despite DUF being well conserved, deleting it is causing this dramatic increase in resistance to antibiotics, which is kind of the opposite of what you would expect, since typically if you see something well conserved, it must be essential. So this suggests that DUF may be playing some sort of regulatory role that we have not yet understood. So in conclusion, we have SMB21269. It's this atypical ABC transporter conferring resistance to a range of antimicrobials. It has this uncharacterized accessory domain, which deletion surprisingly increases resistance. We also know from some other work that this is likely conferring resistance to the secretion of a so far unknown polysaccharide, which is likely acting as providing a barrier effect against antibiotics entering the cell. And currently, it's a real big open question on what role this pathway might be playing in the actual life cycle of the bacterium. Thank you.
Let's go ahead and take 30 seconds to talk to our neighbors. All right, let me get you set up and then I'll tell you just 3 things that you need to know. Yeah. Morgan, you are number 6. Yes, okay. Yes.
Well, I was like, you also what else we know?
Is this right? Yeah. Okay. Yes. Okay. So to get to the next slide, just grab it, the present thing. Yeah. So everyone can hear you, and then don't tell me going on, just go on, like it's not. Yeah, it's not right on time. Yeah. So I'll stand right there and give you like a little 100-minute warning, but I'm okay. Perfect. That sounds good.
Yeah.
I love to hear how loud it gets, and then it gets really quiet. So next we have Morgan. Welcome, Morgan.
Hi, my name is Morgan. I'm a first-year master's student in Dr. Silva's lab, and today I'm going to be talking about differential gene expression in a model of autism using TCF7L2 mutant zebrafish. So just to start off with some background, autism spectrum disorder is a neurodevelopmental disorder that's commonly characterized by altered behavioral and morphological phenotypes. Previous research has shown over 100 human genes that are identified as high risk for ASD. So one of the hallmarks for ASD is synaptic dysfunction. And for the non-neuroscientists in the room, just a reminder, synapses are essentially just your connections between neurons. And so what we can see here in the images is a projection from a neuron of an ASD patient and a control. And what you see is a much higher synaptic density in ASD, showing that different morphological phenotype. My lab generally studies glial cells, and my project will be focusing on microglia. These are the predominant immune cells in the brain, and as you can see from the chart, they have a lot of different functions that are vital to brain function. One of these functions is their ability to interact with synapses and monitor their function and formation. They also have the ability to eliminate synapses, and that generally occurs during development and during disease states. Our gene of interest is TCF7L2. It's one of those genes that was identified as being high risk for ASD, and it's part of a signaling pathway called the Wnt beta-catenin pathway, which is a canonical pathway in development, and it regulates gene transcription. So this specific gene encodes a protein that is an effector protein on beta-catenin in the nucleus, and it's expressed by both neurons and microglia, which is very important as that is our cell of interest. So given what we know about these genes of interest and microglia, our question is, what are the differential genes expressed by microglia in the absence of TCF7L2? To answer that, we did something called single-cell sequencing analysis that essentially allowed us to look at the transcriptome of every cell in whole brain samples of our mutant zebrafish. And once we did that, you get a lovely chart like this, where essentially each of these points represents a cell, and they're clustering together based on how genetically similar they are. Through further analysis, we're actually able to identify each of these clusters based on their cell type. And so what we went ahead and did is we found our microglial clusters so that we could subset them and do further analysis only on this cell type. So the next analysis we did is something called differential gene expression. And essentially what that means is we were looking at the gene expression in our mutants and comparing it to the wild type and seeing what was different. So this volcano chart basically is showing all of our differentially expressed genes and how significantly they were expressed. The y-axis is showing us our significance. So the higher up they are, the more significantly different they are. The farther to the left, the more upregulated, farther to the right, downregulated. So once you have this, you can see some of the top most significantly differentially expressed genes, but it doesn't tell you a ton about necessarily what functions or cellular things are going on in these cells. So that's where our gene ontology analysis came in. And that's what this bar chart here is. It basically allowed us to look at all of our differentially expressed genes and see what cellular functions and pathways were particularly impacted. And what we found were some of the top 3 functions were neuroactive ligand receptor interactions, cell fate commitment, and GPCR ligand binding. These were all affected by the deletion of our gene. So some of our next directions, this leaves us with a lot of ways we can go, but we're going to be looking into some of these most highly differentially expressed genes and trying to find out what are their functions and what role could they potentially be playing in these ASD phenotypes.
Go ahead and chat for 30 seconds. Next we have Jacob. Jacob. Everything to move forward. And then the mic is and then I'll sit there when you're about my Yeah. Okay. Okay.
Thank you.
Next, we have Jacob.
Hi, my name is Jacob Woodall. I am a master's student in the Amphibian Conservation Lab, and my PI is Dr. Vance Friedenberg. And I am going to be presenting you today on my project, Disease Dynamics in the Western Spadefoots. So a little bit of background to start. The disease we're going to be discussing is amphibian fungal disease, also referred to as chytridiomycosis. This disease is caused by the fungus Batrachochytrium dendrobatitis, also fortunately known as BD, which is much simpler to say. This fungus attacks the skin of amphibians and can ultimately cause mortality in severe cases. And it has been linked to the decline of amphibian species globally in North America, Central America, South America, and Australia. Now, for the species I work with, the Western spadefoot is an incredible frog. If you have never had the chance of seeing one, today may be your first. The species Spia hammondii is a California endemic. It is found in the Central Valley down through the south central coast into Baja California. There are two distinct genetic clades that are divided by the transverse range in Santa Barbara County, and I work primarily with the northern clade. This species is threatened at the state and federal level, and it is a very understudied species, especially the non-larval life stages. For my project, combining the two, I am investigating BD infection prevalence and intensity in the northern population of the Western spadefoot. My project has two components, a historical component where I'm looking at infection prevalence in museum specimens, and a contemporary component where I'm looking at prevalence and intensity in living frogs. I'm hoping to look for differences between life stages, metamorphs, juveniles, and adults, as well as in 5 populations in 4 counties. However, as with any field-based project, not everything goes to plan sometimes. Unfortunately, this last spring was a not great water year, so my project has shifted a bit. None of my sites had successful breeding, so I'm only looking at the life stage differences between juveniles and adults. And my 5 populations have unfortunately dropped down to 1 population, as sometimes happens. But fortunately, there still are some frogs around. And here we can see a size difference between a juvenile frog on your left and an adult spadefoot on the right. For my methods, we go for the historic side, we go to various local institutions, Museum of Vertebrate Zoology in Berkeley and the California Academy of Sciences. And it's pretty simple. We just swab frogs and take some metadata. And here's our team at work. For our contemporary populations, it's much the same. We're conducting nighttime visual encounter surveys for the species, which is a very complex way of saying we go out at night with flashlights and the frog's eyes shine and we find them. They are our stars. From there, we set up in situ processing stations. As you can see here, we do much of the same with the museum specimens. We're swabbing the frogs and taking associated metadata. And it is quite a fun time for us and the frogs. We keep finding them. Once we get our samples, we take them back into the lab. We do a simple DNA extraction, and then from there, we run a real-time quantitative PCR to determine if our samples are positive or not. And this is our favorite machine in Hensel Hall. This is the ABI Step 1 qPCR machine in the basement. And here's a sample output from that process. So this graph has a lot going on. And there's some weird things in it as well. Our yellow line is our threshold for positive samples, and what we're looking for is amplification in the qPCR. So if our frogs have the fungus, our probe will attach to that segment of DNA, it'll get cleaved off, and then it'll fluoresce. And the machine is measuring that fluorescence. The samples that are curving up are our standards, which look beautiful for standards. The farthest left ones in red are highly infected or our high infection level standards, medium infection level standards, and then our low standards. The purple line kept me up at night. This was just a mistake in the process. We reran that sample and it came back negative. And so far, for the 100 samples that we have processed, all of our frogs have been negative for the disease, which is fantastic for the frogs and a little stressful for my project. So thank you very much to the department, and thank you very much to my advisor, Dr. Van Spierenberg, and to you all for being here.
Right, so we know the drill, chat for 30 seconds. Yes, okay, so let me pull your slides up. Number 8.
Yeah.
Right, Jaden, so 3 things. To go forward, this is the button. Okay. This is your mic, set it up close so everyone can hear you. And then I'll be sitting there when you have about 30 seconds left onto the ones. We're good on time, so we go over. Okay. Okay. Okay, thank you. Bye. I didn't have to do the bell. Welcome, Jaden.
Thank you. You guys are all very excited for this one. There is no bell. My name's Jaden. I'm in the Galapagos Research Lab, so I'm with Dr. Chavez and Dr. Dumbacher. And I'm going to talk a little bit about the paleobiology that we do in the Galapagos. So we're going to start with a little bit of context. And the Galapagos is an island archipelago that remained relatively untouched until very recently in 1535, where it was used as a rest stop for fishermen and pirates, and they would exploit its natural resources, mostly the giant tortoises. I've heard they're very delicious. I will trust their word on that. But with exploitation of the islands, there was a lot of invasives that likely were introduced repeatedly. And one of the biggest invasives is going to be livestock, which was intentionally introduced as a self-sustaining food source while they were out at sea. And so just a little bit of stats, there's about 1,700 introduced species. Some of the biggest ones that cause issues are rats, cats, dogs, and goats. Introduced animals lead to significant declines in native and endemic species. And since the Galapagos experiences so much tourism, the biggest threat to the archipelago is going to be human impact because of the infrastructure that's needed to support tourism. And so my work is going to deal with the invasive. So our goal is to figure out the historical species assemblage of islands in the Galapagos before humans arrived. And we can do that by looking at paleomaterial that we're finding in the lava tubes that offer evidence of past biodiversity. And so we're finding subfossils, and they will be analyzed using morphological identification. And the results from this will inform restoration and translocation efforts by our collaborators at the Galapagos National Park. And so this is a picture of me from this past summer in a cave by a sand pit. And then I put a couple examples in here of some of the materials that we found. So the one on the furthest side away from me is some land iguana remains that we found in the caves. And then if anybody knows anything about Galapagos, the other one is a Darwin's finch. And you can see that there's plenty of bones in the matrix there. And so what we were able to do this summer is we were able to collect about 2,500 fossils from a cave system on Santiago Island, which is an uninhabited island. And we were fortunate enough to be able to export them, a small sample of them, back to the California Academy of Sciences, where we can use the scientific collection to do a comparative anatomy study and see what species are historically there. And so my thesis is going to focus on writing the descriptions on how you can identify the bones from each order, family, and genera. And this is a picture of the cave that we ended up having to rappel into. That's Jack on the line right there. And those are the park rangers up at the top that were not enthusiastic about rappelling. And so I just wanted to share a couple more pictures. Ultimately, we're using cave bones to determine what species were present on the islands in the Galapagos before humans disrupted the ecology. And it is important because the local people want to restore it to as close to the wild places it's been.
And so the picture with the giant tortoise in the mud is from the top of Santiago. And then this picture where I have the backpack on is actually that peak up there. And then this is the rack I got in my tent when I didn't zip it up.
Okay, thank you guys.
Right, so go ahead and do your 30-second chat.
I have my time.
Oh, wonderful, okay.
Just because I like to look at it.
Yeah.
And then I'm really super confused to figure in like when I got here this morning.
Oh,
I uploaded it and changed it, so
Okay,
it's just
I just,
like,
mhm.
okay.
Do you want to double check? Do I have to refresh or is it there?
There it is.
Okay,
There.
wonderful. Oh, you have a PDF, right?
Yeah.
Okay, let me see how it's gonna open and then.
If I need to send it as a
Let me see if I can,
slides, I can do that.
there are ways, do you know how to present from slides?
I mean, I can send it as just like a Google Slides presentation. That's better.
Does that work?
Yeah.
Everybody else out there, it's like a reading. No, they just dragged it in, you wanna download it? I did download it. Oh, so I should download it as a, oh, yeah, yeah. Oh, okay. Okay, so I'm gonna close this and then I'll go back to that file. It's the one at the top. That's wonderful. Kind of messed up something. Is there anything you can change?
Just like it's supposed to say like Brad Lightning's on.
Yeah. Okay.
Sorry.
I think everything else looks okay, right?
Yeah, I don't really see any over.
Oh, that's fine. Okay, and then, so I'm gonna tell you 3 things. To move forward, you're just gonna press this button here. Have this mic, we're gonna close these up. Then you have your several bells. Yeah, okay. And our last speaker is Sierra. Welcome, Sierra.
Yes, we made it. This is the end. Alrighty, so I'm gonna get started. So my name is Sierra, as Kathy said, and I am in Dr. Derek Grew's lab, and I study hummingbirds. He actually presented 2 weeks ago. And actually my research is a little bit different than all the stuff that he presented on. I'm looking at, thermoregulation and thermodynamics and stuff with the birds. So my project is entitled Body Size Changes in North American Hummingbirds Following Range Expansion. And I am actually studying museum specimens for my data. So to summarize sort of the question I'm trying to ask, as well as to give you guys a little bit of background, I'm studying 2 species of mostly West Coast North American hummingbirds, the Anna's hummingbird and the non-migratory subspecies of the Allen's hummingbird. So Anna's hummingbird, these guys are everywhere, all over the place, especially around the city. You hear them shouting at each other all the time. They have the green body with like the purplish reddish face. And then the Allen's hummingbirds, the migratory and the non-migratory subspecies, they usually have the greenish body with the orangish face. And I'm essentially looking at these 2 species and their relatively recent range expansion, and subsequently their exposure to new climates in these different ranges that they've expanded into in the last 60 years or so, and the expected morphological changes that can follow these 2 ecogeographical rules that I'm going to go over, Bergman's rule as well as Allen's rule. So for the first ecogeographical rule, Bergman's rule, essentially it states that populations in colder climates
tend to have a larger body size compared to populations in warmer climates, which tend to have a smaller body size. And a really great, convenient example that I like to talk about are penguins. So the emperor penguin is the largest species of penguin, and they live in the South Pole, where it is very, very cold. And then as you get up closer to the equator, the species of penguin gets smaller in body size. For Allen's rule, it is looking more at limbs. So populations in colder climates tend to have shorter and thicker limbs, where populations in warmer climates tend to have longer and slimmer limbs. So my example for you guys to look at is looking at different species of hares. So the Arctic hare is very short and squatty. It has short legs, thick legs, and short, thick ears. And then we have desert hares all the way at the end, and they have very long ears and very long, skinny limbs. And essentially, both of these ecogeographical rules have everything to do with thermoregulation in these different environments. So as I mentioned earlier, the Ennis hummingbirds experienced a relatively recent range expansion starting around the 1960s. Historically, they are native to San Francisco Bay Area, all the way down the coast into Baja California. And you can see on that figure there, the colder temperatures indicate the historic range. So from the 1940s through the 1960s and 70s. And then once you get into the warmer colors, the yellow, orange, and red, that is where you get into this expanded range. And you can see they have moved all the way up into the Pacific Northwest, British Columbia, and even into Alaska. So it's pretty far north for a little guy. Previous studies have shown that they're actually following human settlements. So humans plant trees and flowers that they're able to find food and habitat in. They're able to nest in these locations. And they also get to have access to supplemental feeders. So humans like to put out hummingbird feeders. They're so cute, we see them. So that helps them move into these areas that they haven't been living in previously. Similarly, the non-migratory subspecies of Allen's hummingbirds have established a population in the Los Angeles basin. So in the red circle, I have the Channel Islands circled. And the non-migratory subspecies are native to the Channel Islands, and they have moved over within the last 50 or 60 years. And they were first noticed on the Palos Verdes Peninsula. Relatively in the 1970s, they could have been found or could have started in the 1960s. They were first recorded in the 1970s. And once again, like the Anna's hummingbirds, they're constrained to these urban areas. So mostly in the urban and suburban areas of the Los Angeles basin, closely in association with humans and human settlements, the planted trees, flowers, and the supplemental feeders. So I hypothesize that Anna's hummingbirds in the colder climates in the Pacific Northwest and British Columbia are larger and have shorter, stockier appendages over time following the expansion north. And the non-migratory Allen's hummingbirds in the warmer climates of Southern California, the Los Angeles urbanized basin, are smaller in size and have longer, slimmer appendages compared to the island populations on the Channel Islands. So for my data collection, I'm looking at museum specimens, like I mentioned, historic data, and I'm a big proponent of museum collections. It is a mass of data that we're able to access, and it's really important if you're looking at things, changes over time. It can also inform how we might be able to make predictions about the future. So the measurements I took from the birds were mass at the time of collection, if it was available, the bill length, the bill width, the tail length, and the wing chord, so all the appendages. And I visited 4 museums in my data collection, which is almost finished. I need to go back to the Cal Academy, but I visited the Cal Academy, the Museum of Vertebrate Zoology at UC Berkeley, the Natural History Museum of Los Angeles County in Los Angeles, and then the Burke Museum in Seattle. I also took, in addition to my body size measurements, I took some camera images of the faraway museums because I don't want to have to visit those places again and spend all that money. So I took top down as well as side profile pictures of the birds, as well as having a ruler in frame so I can take any supplemental measurements digitally if I need to. Like I said, I'm still finishing up my data collection as well as getting into my data analysis. So I don't have any results for you, unfortunately, but I do have some expected results. So Allen's hummingbirds, the non-migratory subspecies, serve as a counterpoint to Anna's hummingbirds, and their body size changes as they've expanded in these two sort of opposite temperature regimes. And I'm interested to see if there's an interaction between Bergman's rule and Allen's rule, or if both are playing a significant role in the way that these birds are changing. And thank you so much.
So now I want to invite all of our speakers to come up. So we have Lydia, Michael, Gabriella, Billy, Clara, Morgan, Jacob, Jaden, and Sierra here.
Jill.
Oh, So now that we have them up here, first, I want to give you guys all a round of applause. And also say thank you to Dr. Sway for organizing this. We hope to see this every year, hopefully. So now we're gonna open up questions for faculty. We'll give faculty a couple of minutes to ask questions, and then faculty will be dismissed and we'll have an opportunity for students or prospective students to ask questions. I'm also gonna be monitoring the chat, so bear with me as I go back and forth. So let's go ahead and open it up for faculty. You can ask general questions for the panelists, or if you want to be specific, we can also do that. Do we have faculty? I haven't seen any. Yeah, Beth.
By the way, really nice. It's so cool to see the breadth of talks and research that's happening across the department. So thank you all for doing this. I had a question for Sierra, since that was the last talk that just came up, which was, so the expansion of these two species in habitats, is it generally just because of, like in the picture here, because of feeders, or is it more because of the plants that are being grown in people's gardens?
As far as I know, it's a combination of both things. I hope I'm speaking loud enough for the people in the back to hear me. Okay, thank you. As far as I know, it's a combination of both things. I think that a couple of the papers I've read that helped influence my research was looking at eucalyptus and planted eucalyptus, especially along the West Coast, is one of the major plants that contributed to this expansion. The other thing I didn't mention, because I didn't have time, was also looking at Anna's hummingbird specifically. They've had a really large population boom in the Bay Area and into Central and Southern California. So that was also a contributing factor that contributed to their expansion north, is that there was just a lot of hummingbirds in one area and they needed somewhere to go. And it just so happened that there was food and habitat for them in these places.
Jonathan, a question from Jonathan?
Question for Jaden, nice presentation. How do you tell when the bones you find in the cave got there?
That's a great question, and the easiest answer is carbon dating. But until you can figure out what they are, and if it is something that has gone locally extinct on the island, carbon dating is kind of expensive, so it is a little outside my project at this time. But I think overall it will probably be incorporated further on.
Last chance for faculty. All right, wonderful. I'm good. Yeah, Dr. Slay, go ahead.
So yeah, great job, everyone. That was so wonderful to hear all of your research. I have a question for you, Jacob. With the frogs, like, are there any, is there evidence of declines in the Bigfoots that you're working on? And are you surprised that you're not finding infection, or can you elaborate on that?
Yes, so the species is really complex. There are declines in the Central Valley that are largely linked to land loss. The one population I study is currently in a boom cycle, we believe, due to the previous year's rains. We're seeing more adults than anyone has ever seen at the site. But overall, in a population decline, I am surprised to not see chytrid on the spadefoots or other frogs present at the site. A previous study done, or previous sampling done by the USGS at the site in 2019 did find positive frogs for separate species. So we were anticipating to have chytrid. The site is also an active motorcycle park or dirt bike park. So there's a heavy amount of traffic, which doesn't seem to bother them, or maybe affect it.
All right, any last questions for faculty? All right, faculty, thank you for coming. You are now dismissed. And while all our faculty make their way out, I want to invite our students to spend maybe 30 seconds to a minute to chat with each other to come up with a couple of questions. And then I'll go ahead and open it up for students. So it's going to get really loud. Okay, I can do that. Thank you. Yeah. I love how loud that got. So now we're gonna open it up for questions from the audience. And then I'll leave maybe about 7 minutes at the end, so that if you have a question, you have time to talk one-on-one with our speakers today. So the first thing we're gonna do is I'm gonna wait for 3 hands. Oh, nice. Okay, that was fast. All right. 1, 2, 3. Sam, we'll start with you. I have a question for Gabrielle. So I know, like, how do you all the morphology where they're using like gauze, and it's stirring as well. And so, there have been like tests where they've had like machines that collect a composite sample, so it'll like every like 15 minutes it'll collect a small amount of water. And then that's a small sample, and then we have people who have done like more swab type methods, and then we have people who just do like 1 graph sample at one time, and they compare those, and they've seen that the like swab type method, that is like similar to the tampons, that is similar to composite samples and not the time samples. Great question. The question in the back there.
Question for Jacob. Great. I have a question. Like, do you guys need blood samples from frogs for your PCR and for gene extraction?
Do we need more?
Blood samples
Blood sample?
for your gene extraction and for PCR.
Blood. Oh, no, sorry. No, we don't need blood samples. So Chytrid only infects the skin, the epidermis. So if the spores are present, they'll just be present there. Sorry, I'm not using the mic. I'm kind of a loud talker. So no blood, fortunately, which I'm very happy about. It's a very non-invasive process. And they love it, I swear. We keep finding them. Like, and there's certain individuals, like there's a one-eyed frog, like, yeah, they love it.
And then we had a question. Yeah.
First of all of you did amazing, like really great job for all of you. It was really awesome to learn about all of your different projects. I have one question, one for Sierra and one for Jacob. Jacob, since you already have my guy. You said that you had 5 different areas that you were gonna take frogs from, and that I think one place ended up with frogs. Now, are you saying that's because due to the lack of rain, but the breeding ground wasn't there, or are you actually worried that it's gonna fully be extinct by the time your project finishes up?
That's a great question. So 2 of the sites did have frogs. They just had extremely small sample sizes, 1 frog and 4 frogs. The difficulty with the species is they're complex. So sometimes they can be explosive breeders, but we've also found them like through July at our current site. So I have absolutely no idea. People don't study the adults. So most of my sites I found were found because people had found tadpoles there. And several of the sites, we were the first people to find adults in 20, 30, 40 years. So it was a huge complication with pastoral species, and then just access to those sites as well. They're further away, so it's harder for us to go out there, whereas the Carnegie site, we were surveying weekly. It's much closer. So even if we have a bad week, it's fine, like go out next week. But when your site's 6 hours away, it's really difficult to go out there every week. And so it's just potentially missing that frame. Part of the other thing I didn't present on is I've also created a model with my project with the data that I have about the environmental covariates of terrestrial activity. And I'm hoping to start the next spring, use that to guide the surveys to see if maybe like that works or if the model lies, which they tend to.
Sarah, and I'm microbiology with Gabby and Michael, so I don't have a lot of knowledge about animal physiology and how that works, but you mentioned using historical samples. Are you also using live samples like wild type hummingbirds, and how are you catching them and measuring them as well if you are doing that?
Yes, the answer is no. I'm not measuring any live birds, just museum specimens which are dead. But the Cal Academy as well as the Museum of Vertebrate Zoology, actually all of the museums have pretty relevant collections up to 2020. And I didn't put this in my slides, so I didn't have time, but I'm only measuring birds collected from the 1960s to the present for the Anna's hummingbirds, and then the 1970s to the present for the non-migratory Allen's hummingbirds. And I have a pretty consistent range as much as I can get with historical specimens. Often you can't go back to the 80s and collect birds from then. But especially as you get into closer to right now, collections became more popular and there's definitely more. So there's a lot of specimens that were collected within the last 10 years or so. And then whatever's left for the other decades, I've measured all of those that were made available to me. So I guess your question is like why I didn't use any live birds, or yeah, just because the museum specimens are easily accessible and they're very similar or assumed to be very similar to the ones that are alive right now if they were only collected like 5 years ago.
I'm assuming you didn't have to get a permit to go to the.
No, I did not. I just emailed the curator of birds and I show up.
We have time for other questions.
Yeah. Thanks again. It was great talk from all you guys. I have a question for Clara. I know you said SMB 21269 had the resistance to these antibiotics, and I saw you put a various level of like nanoliter concentration.
Yeah.
Correct me if I'm wrong. Like
It was nanograms per mil. It was just,
okay.
it was just for one antibiotic. Yeah.
Okay, so I was just wondering, like, how did you come up with this concentration, and how is it related to the like impact in the scope of public health or environmental factors?
So for the concentrations, it's basically, I'm just doing 1/2 serial dilutions, and just on a 96 well plate until I can find a range where I can see the minimum inhibitory concentration for both of those genotypes. As far as public health wise, I mean, it's always possible for this resistance pathway to be acquired by pathogens. However, we have not found this in any current pathogens. In fact, there's a very closely related human pathogen to this bacteria called Brucella, where it's completely absent, but then it is present in even further away bacteria. So right now, it's not clear on what the impact would be on human health.
Oh, hi, thank you everyone, and my question is for Sierra. So I've seen with Bergman's and Allen's rule that a lot of time it's like interspecies comparison. Has there been other studies on like species we can follow with patterns?
Yes, there's a couple. Again, I didn't have time to include this, but there's also a lot of relatively recent research on the hummingbird bill as a thermoregulatory tool. In the instance that these birds are in a warmer versus cooler climate, we would also see it's kind of, they trend similarly. So if the bird, for example, doesn't need to do as much heat dumping through the bill, then it could be shorter, doesn't need to be as large. And then if it's in a warmer area, it might need a larger bill to do more heat dumping. So there's a lot of things that are influencing the size of these birds, and that's definitely something that we considered. And that's probably something for like the future to look at. With Bergman's rule and Allen's rule, there has been some looking at also birds, mostly shorebirds, I think, or seabirds, looking at how Bergman's rule is acting on these birds.
All right, any more questions?
Yeah.
All right, so what I'm gonna do now is invite you up. If you have any questions for our speakers, a one-on-one, come up here and ask questions. I'm also gonna put our sign-in sheet, so A through H is gonna be on this side. And J through Z is gonna be on this side. And then the last thing I wanna do is give our speakers another round of applause.
I should probably sign in.
Yeah, the questions don't have to just be about the project. You have questions about grad school, touching bra,
Yes.
licking bra.
Answering this question.
Yeah.
Yes. Inspired.
Oh, you didn't. You really
Should I just type it?
Okay.
It's pretty loud in here, okay.
And there you go.
Probably not the correct path.
Okay. Really? Okay, yeah. I look at you. This is like.
I'm gonna use the word.
Project
I have looked at some of the, like I've done the whole Spain.
Oh, this is Avery.
Samples. But we weren't getting that good deal. Mostly we get like all the hosts, and we only got one host. And then also.
Bye, everyone.
Yeah, we basically measure about like Zeus makes